Welcome back. Transport in plants explains how water, minerals, and the products of photosynthesis move over long distances inside a plant, entirely without a central pump like a heart. In this video we'll cover water absorption, the ascent of sap and transpiration pull, apopul, and translocation via phloem. Let's begin. Water enters a plant primarily through root hairs, thin, elongated extensions of root epidermal cells that dramatically increase the surface area available for water absorption from the soil. Water moves from the soil into and across the root by osmosis, flowing from a region of higher water potential in the soil to a region or lower water potential inside the root cells, and it can travel by two distinct pathways once inside the root. The apoplast pathway involves water moving through the interconnected, non-living cell walls and intercellular spaces, without ever crossing a cell membrane, and this pathway offers comparatively little resistance to flow. The symplast pathway, in contrast, involves water moving from cell to cell through the living cytoplasm, passing through tiny channels called plasmodesmata that connect adjacent cells. Critically, both pathways must eventually merge and pass directly through the endodermis's living cells, because the endodermis's Casparian strip of waxy, water-impermeable band completely blocks the apoplast pathway at that specific point, forcing all water to cross a cell membrane there. And this is precisely how the plant maintains selective, controlled entry of water and minerals into its vascular tissue. Once water reaches the xylem at the center or the root, it must be transported upward, often to great heights in tall trees, and this movement is explained by the cohesion tension theory, currently the most widely accepted mechanism. Transpiration, the continuous evaporation of water from tiny pores called stomata on the leaf surface, creates the negative pressure, or tension, at the top of the xylem column, and this tension pulls the entire connected water column upward from the roots, much like sucking liquid up through a straw. This pulling mechanism only works because water molecules exhibit strong cohesion, hydrogen bonding tightly between water molecules themselves, allowing them to be pulled upward as one continuous unbroken column without the column snapping under tension, and water molecules also show adhesion, sticking to the walls of the narrow xylem vessels, which helps counteract the downward pull of gravity on the water column. This entirely passive mechanism requires no metabolic energy expenditure from the plant itself. It's driven purely by physical forces, evaporation, cohesion, and adhesion, working together. Stomata are microscopic pores on the leaf surface, each flanked by two specialized guard cells that regulate the pores opening and closing, and this regulation, called stomatal movement, is primarily controlled by change in the guard cells' turgor pressure. When guard cells actively accumulate potassium ions, water follows by osmosis, increasing their turgor pressure, and because guard cells have unevenly thickened walls, thicker on the inner side facing the pore, this increased turgor causes them to bow outward, opening the stomatal pore. When potassium ions and water instead leave the guard cells, turgor pressure falls, and the guard cells straighten, closing the pore. While transpiration serves essential purposes, driving the ascent of sap, cooling the leaf surface through evaporative heat loss, and maintaining the cell turgidity necessary for structural support, it also represents a significant, sometimes problematic, water loss for the plant. So stomata typically close under water-stressed conditions, often triggered by the plant hormone abscisic acid to conserve precious water at the direct cost of temporarily reducing carbon dioxide uptake. Photosynthesis. While xylem transports water and minerals unidirectionally upward, phloem transports the organic products of photosynthesis, mainly sucrose, bidirectionally from a source, typically mature, photosynthesizing leaves, to a sink, any region actively consuming or storing food, like growing roots, developing fruits, or storage organs, and importantly, which organ acts as a source versus a sink can actually change over the course of a plant's life depending on its current metabolic needs. Translocation process is explained by the pressure flow, or mass flow, hypothesis. At the source, sugar is actively loaded into the phloem's sieve tubes, using energy, which sharply lowers the water potential inside the phloem there, causing water to enter from the adjacent xylem by osmosis and raising hydrostatic pressure at the source end. At the sink, sugar is unloaded and consumed, or stored, raising the local water potential inside the phloem, causing water to exit, which lowers pressure at the sink end. This pressure difference between the high pressure source end and low pressure sink end drives a continuous bulk mass flow of the sugar-rich solution through the phloem, from source directly to sink, entirely along a pressure gradient rather than an aquavake pumping mechanism. Let's recap. Water enters through root hairs and crosses the root via apoplast or symplast pathways, both forced through the endodermis at the Casparine strip. The cohesion tension theory explains how transpiration pulls water up the xylem passively, relying on water's cohesion and adhesion properties. Guard cells regulate stomatal opening via trigger pressure changes driven by potassium ion movement balancing. Transpiration's benefits against water loss. And phloem translocates sugars bidirectionally from source to sink, driven by a pressure gradient created by active sugar loading and unloading, as explained by the pressure flow hypothesis. That's transport in plants. Keep xylem's passive, upward-only cohesion tension mechanism, clearly distinct from phloem's active, bidirectional pressure flow. See you in the next video.